Hello friends, welcome to my channel CM's Biology for a new video. So here today we are going to discuss a very important topic which is very interesting and sometimes makes puzzle in the mind of the students. They get confused about these things. So let's discuss about this very important connection, that is the connection between the parents and offsprings, and the variation and heredity. What are the similarities, why the similarities is found between parents and offspring, and why the dissimilarities occur. So what are the cause of dissimilarities and why similarities occurs between parents and offsprings. So today we are going to discuss about linkage and recombination. It is very important for the 12th standard students. So focus on this video so that you can easily understand at the end. So linkage and recombination, before going in detail, we should know that according to chromosomal theory of inheritance, the chromosomes carry the genes and the number of genes is more than the number of chromosomes. And remember, we know it very well that the characters are present in the gene, and that is the reason why genes carry the characters from parents to offsprings, and the number of genes is more than the number of chromosomes. So a single chromosome carries more than one gene. Okay, so first look at this. The linkage was first demonstrated by Watson and Punnett. Punnett you know very well in the while doing the Punnett square in monohybrid and dihybrid class. So Watson and Punnett together demonstrated the theory of linkage, and in 1906 he conducted his their experiment together on sweet pea or the Lathyrus odoratus. So he used the sweet pea plant, Watson and Punnett, to conduct the experiment of linkage. So he found something different from Mendelism here. So Watson termed his result as coupling and repulsion hypothesis. We will discuss about the coupling and repulsion hypothesis in this video also. So remember, first let's go towards linkage and how the linkage is related to heredity. So now let's see what are the characters of linkage, why linkage occurs, and what are linked. Let's talk about it. So genes found on different chromosomes are unlinked. So when the genes are found, one gene is found here and another found here in a different chromosome, these are called unlinked genes, and they are found in different chromosomes. And when they are found in the same chromosome, they are called linked genes, and when they are in different chromosome, they are called unlinked genes. Okay, clear? So remember, distance is an important factor. So when the distance is large between the genes, the chance of recombination is large. When it is called linked genes when the genes are present in a single chromosome, but when they are far away from each other, the chance of recombination is more, means the chance of linkage is less. So when the distance is large, chance of recombination is large. Distance large, so chance of recombination is also large. Okay, when the genes are far away in a single chromosome, far away from each other, there is chance of crossing over, which results into recombination or appearance of new characters different than the parental character. Okay, so chance of crossing over is also larger. Chance of crossing over crossing over is also large. Okay, so when the distance between the two genes is more, the distance is more, at that time the chance of recombination is more, and accordingly the chance of crossing over will also increases. But when the distance is small, means the two genes present in a single chromosome and they are close to each other, at that time when they are closely placed or the distance is small, the chance of recombination or crossing over is small. So here the chance of recombination was large and crossing over is also large, but here chance of recombination and crossing over is less. Here it was large. Okay, so when they are closely placed, the genes in the chromosome are located close to each other, at that time the chance of crossing over or their separation is very less, so they are called linked genes. When they are distantly placed, they are called unlinked genes, or when they are present in two different chromosomes, they are called unlinked genes. Clear? So linked genes and unlinked genes. So when the genes on same chromosome, they are linked, and when they are placed very close to each other, there is less chance of recombination. Means when they are very close, very near to one another, there is least chance of recombination. So recombination cannot occur when the genes are very close to each other, because they cannot separate from one another during crossing over of the non-sister chromatids of homologous chromosome. Clear? So when crossing over do not occur, they do not separate, and sometimes crossing over also occurs, but they cannot separate because they are linked very close to each other. So linked genes are placed close to each other, unlinked genes are placed far away from each other or in two different chromosomes, that results in crossing over or recombination. So when the distance is large, or the genes are located far away from each other, at that time they cannot travel as a whole. In this case, they can't travel as a whole. Okay, they together cannot travel, so that more percentage of recombinant can be produced. More percentage of recombinant are produced, and less percentage of parental character is found. So less percentage, so I am giving arrow downward, less percentage of parental character is found, and more percentage, I am giving arrow upward, more percentage of recombinants are found. But here in this case, when the genes are linked, the distance is small. When the distance between the genes is small, at that time they can travel as a whole. So in this case, here they means the genes can travel as a whole, so they together can travel because they are very close to one another. So the percentage of parental character is more. So more percentage of parental character here, and less percentage of recombinant. So in this case, as the genes are linked together, situated close to each other, so the chance of their arrival to the offspring is as a whole. So together they can come to the offspring to represent their character. So parental character is found in higher percentage and recombinants are found in lower percentage in the case of linked genes. If we will summarize this whole thing, I can write down like this. So concentrate here, note down these important points. So from these details, we can summarize the idea of linkage and recombination. Linkage crossing over results into recombination. So how we will summarize them? When genes are far away, at that time the percentage of recombinant is high. So we can write down genes far away. At that time the percentage of recombinants will be more. Okay, so recombinants will be more means the parental character will be less, means percentage of parental character is less. Okay, so from this we can say chance of crossing over, as the recombinants is more, so the chance of crossover is also more. Okay, because these two are connected with each other. Clear? Next, when genes are close, genes are far away in that case. In the second case, when genes are close to each other, what will happen? Percentage of recombinants will be less. Percentage of recombinants will be less due to the less crossing over. So the chance of crossing over will be less. So the chance of crossover will be less. But in this case, the parental character or the percentage of parental character will be more, as the genes are closely situated, closely located. So these are linked genes. And in this case, these are unlinked genes. Okay, so in case of unlinked genes, the less parental character is seen. In case of linked genes, parental character is higher in percentage. So from these we can also write down the linkage frequency is proportional to 1 by distance between genes. Write down linkage frequency. So how we will calculate the frequency of linkage? Linkage frequency is proportional to is related with what? The distance between genes. So when the distance between genes increases, the linkage frequency decreases. Distance between genes. Okay, and we can also write down the recombinant frequency equal to the recombinant frequency equal to, how to calculate the recombinant frequency? Total offsprings or the total offspring recombinant offsprings divided by total offspring. Total recombinants offsprings divided by total number of offsprings into 100. So this is the formula to calculate recombinant frequency. Total recombinant offsprings divided by total number of offsprings into 100. Another point, distance between two genes is measured by centimorgan or cm. Distance between two genes. Distance between genes is measured by Small c, capital M means centimorgan, named after the father of linkage, Morgan, T.H. Morgan, the father of linkage. He found linkage for the first time in the Drosophila, okay, fruit fly. So according to his name, the distance is calculated by using the unit centimorgan, so cm. Next, linkage frequency is inversely proportional to crossing over. So linkage frequency is inversely proportional to the distance between genes, and it is also, linkage frequency is also inversely proportional to what? Inversely proportional to the crossing over. Crossing over. Because when the distance between genes increases, the chance of crossing over also increases. So as distance between genes is directly proportional to the crossing over, so it is also inversely proportional to the frequency of linkage. So these are the fundamentals of linkage and crossing over, how different characters appear in crossing over, due to crossing over, and recombinants are found which are not similar to the parental characters. So this is the basic concept about linkage and crossing over, and what is the result of crossing over, how recombinants are appear in the offspring. So why the deviations from parental character occurs in the offsprings, and what are the different causes, we will discuss again in the next video. And we will discuss about cis and trans arrangement of genes, what is coupling and repulsion hypothesis, and we will discuss two different scenario, one where the genes are located in the same chromosome, and another when the genes are located in two different chromosomes. What will be the percentage of recombinant characters, appearance of recombinant characters, and what will be the appearance of parental characters. So thank you very much. If you like these videos, please subscribe to my channel and keep watching my videos and keep learning. Best of luck.